The topic of my thesis was, can you catch a heart attack? I spent 4 years coming to the conclusion that the way to a man or woman's heart is probably not through their stomach. That Helicobacter pylori was not likely to be a causal etiological factor in coronary disease. But what I learned from that about the need to study at scale for systematic approaches and to try to mix biology with epidemiology is something which has persisted, and that's what I'd like to discuss today. Chronic infections and heart disease was a fever of the mid-1990s. One of our current areas of intense interest is this idea of multi-omics. And essentially this talk is to say that assay technologies seem to be finally catching up with Richard Peto's vision for large-scale blood-based epidemiology. And it's much easier nowadays to make the argument for such studies, largely because finally, after 30, 40 years that Richard's been making the points, the technologies are maturing. So we now have a language to talk about these technologies. We call them omics, to refer to collectivities. The collection of the human genome we call genomics, and therefore, by analogy, the collection of proteins we call proteomics, lipidomics. Someone once said the only omics that really matters is economics, because these things are expensive. What they share in common is that in contrast with static DNA, these are dynamic, potentially modifiable factors, and therefore the hope is that they may lead to some breakthroughs in understanding, perhaps identifying a new LDL cholesterol, or tell us about the mechanistic chain of events that link DNA genetic variation with disease. We've been very good at building DNA maps in the last 20 years, but these maps are somewhat barren. They're not actionable maps because we cannot connect the DNA letters with the diseases through any kind of meaningful knowledge. And that's the quest of this type of endeavor. So these are the types of areas that have been trumpeted as potentially valuable. And of course, the idea is to try to tackle this with some of the inherent skepticism of an epidemiologist and to use Peto's paradigm. Is it really worthwhile, then it's worth doing well or worth doing reliably. So these are some of the things that we really don't know about some of these assay technologies. We don't know really their basic properties, how well they work in populations. They may measure many things, but are they measuring things that are meaningful, reproducible? And I'd like to show you a few slides that suggest that at least for a few of these technologies, we may be making some surprising progress. So in Cambridge, I lead a program, systems genomics programs, if you like, that's trying to combine some of these assays in the same individuals who have been sequenced and linked with health records. And today I'm just going to tell you about the study of proteins. Proteins fulfill the bleeding obvious paradigm. They're the building blocks of biology. When they go wrong, diseases happen, and they're the targets of many drugs. So they seem a natural place to start. And the reason why it's been very difficult to study them at scale, both in lots of people and lots of proteins, is that proteins are a many-splendored thing. So for example, this assay system measures 5,000 proteins, and one of the key things that was difficult that it achieved that others haven't done is that proteins exist over 8 orders of magnitude, so many different, almost a billion-fold different concentrations in the blood. So coming up with an assay system that measures the most minute proteins as well as the really common ones is tricky. This inventive company has come up with these so-called aptamers, single strands of DNA, and they combine, a bit like Richard, both analog and digital properties. So they're analog because they can wrap themselves around proteins, and they're digital because they can hybridize with other strands of DNA. So you get reasonably specific reads of many different proteins in a quantitative way, at least in relative quantities. It's not quite an agnostic platform. As you can see in this case, the proteins have been selected because of known biology and potential therapeutic target interest. So over the last couple years, we've tried to evaluate this assay system, and the results came out last month, led by PhD student Ben Sun in my group, and also principally led by Adam Butterworth. So what I'd like to show you is how we took some of the basic principles of epidemiology that Richard Peto has taught us and try to apply that to an assay system that measures several thousand proteins concurrently. The first question is, are the assays reproducible within an assay plate? And the answer is that for most of these proteins, the coefficients of variation are reasonable. The second question is the concept of regression dilution that Sarah Lewington introduced. What is the year-to-year chopping and changing within individuals of these proteins? And as you can see here, there is a distribution, but many of the proteins fall in this 0.4 to 0.7 range, which is comparable to risk factors that we understand well, like blood pressure or LDL cholesterol. So there are, for the large majority, meaningful results. Do these proteins correlate with known features of physiology or epidemiology? And the answer is, for those that we have good examples, the answer is yes. And then perhaps the type of validation that makes us even more satisfied is this. So let me explain this somewhat complicated slide. Each dot that you're seeing is a highly significant statistical association that connects one letter of the genome with one of the proteins, one of the 3,500 proteins measured by this system. So if you look at the horizontal axis, this is ordered by the genome. So these are the chromosomes, 1 to 22. And then the vertical axis takes advantage of the unique property of DNA that each protein, each gene encodes one protein. So we've numbered the proteins that are encoded by the same genes in the same order. And so this beautiful red diagonal line is telling us that we're finding many hundreds of examples where the gene, a variant that's in or near a gene, encodes its own protein. And of course, this is a validation that the assay system is working. The blue verticals tell us something interesting. They tell us about hubs of protein machinery. So some of these hubs are already well known, complement factor H. So for example, responsible for many biological cascades, protein cascades. Other ones are perhaps more surprising. And then the side-to-side scatter tells us perhaps potentially about new biology, associations between proteins and protein systems that weren't known before. And one of the potential advantages of this type of approach is that it may help tell us something about etiological factors in disease. So here's one example. It's an enzyme that's produced by a white cell called a macrophage. It's called MMP12. It's a bit of a mouthful. But it's under investigation in certain pharmaceutical companies as a possible inhibitors to it as a way of treating chronic lung disease. One of the findings from this protein assay is that we're able to build these types of allelic scores where we can look at, across a gene, many different independent variants to try to create something of a genetic mimic of drug effects. So in this case, we can explain about 15% of the variation in blood levels of this enzyme with about 12 or 15 independent genetic variants. And this works a bit like a dimmer switch. So these genetic variants are associated with lowering, with higher levels, these with lower levels. And then when we project this information into separate samples of cardiovascular disease using something that Zheng Ming alluded to before, a technique called Mendelian randomization analysis, we see this surprising result that higher levels seem to be protective of disease. And we see this not only for coronary disease, but we see it for stroke, suggesting that those companies that are developing inhibitors to these agents might want to be careful. So as a vignette. So Valerie's asked me to stop, so I think I'll stop there by basically saying that assay technology is finally catching up with Richard Peto's vision, and I've been very lucky to have excellent PhD supervisors. Thank you very much.